Laminar airflow, you need also this thing sterile. These are filter papers. These are regular papers that needs sterile. So you first autoclave them and then use. So let's say we are working with this, okay? Cut your plant material. So I have now few leaves. Try to take, not to take petiole part for this thing. Try to take only, because sometimes petiole parts do not contribute towards regeneration, and your leaf there will be a regeneration from your leaf part. So that you need to first optimize before you go for the plant transformation experiment. So you need to see from which part, from the petiole it is the new plant is regenerating, or from any other part. So you need to take that part on. If it is regenerating from, let's say, internode, you have to work with the internode. So let's assume that it is regenerating from the leaf. So we are taking leaf. So you need to make small pieces of the leaf. Let's say this is already small. We can take two this thing. Yeah, that's what I also think. They can come around.
But in the lab, this has to be done in aseptic.
Yes, but here also it's not, because
But
there is
people, they are in the first year and they don't know about the explant, they don't know about what is callus.
okay.
Maybe node, internode, anybody know? Node, internodes?
Yes, sir.
What is this node? What is a node? Anybody? So they are in
That is
that
specific.
level. So they will be, anyhow, they will be given chance for hands-on experience. But what I'm feeling is, since they don't know, the backsiders already they started watching codes. So the point is, they don't know what they are doing here.
Yeah, no solution. So after cutting, you just collect them and put in a agro solution for, let's say, half an hour. I can circle. You can see like this. Okay, you just need to put them in a shaker. This is agro solution, and with your leaf material, the explant material. So this explant is going to regenerate into a new plant. How? Through the plant tissue culture process. But before that, your agrobacterium will transfer your gene of interest into the plant. So the new plant that you will get it will be or will not be having your gene of interest. That portion can be screened with the selection method. Okay? Because in the plasmid that we have placed inside the agrobacterium, that also not only have your gene of interest, but also has a kanamycin resistance gene. So plant has also acquired the kanamycin resistance now. So what will happen if you put the plant in kanamycin containing medium? That plant will survive. But who will not survive? The plant without having that kanamycin resistant gene. Which plant will not have that kanamycin resistant gene? Which is not transformed. Okay? Because during transformation it happens that some of the cells will get the plasmid, your gene of interest, some of the cell will not. And as I told you, sometimes it also happens that the kanamycin, the kanamycin resistant gene, even if it is not there inside the plant, it can survive to the lower concentration. So you need to give more and more of kanamycin, so that to make your selection stringent. Right? So all these procedures are after like 21 days or after one month you will start the selection and all this process. But before that, where we have to start with the transformation, you just need to cut the leaf and put it in an agro solution like this. Now the laboratory must have a shaker. Place it on a shaker for half an hour, 80 rpm, 80 to 100 rpm. You need to shake it, so that during that process the agrobacterium will find the space in between the cells. We have cut the leaf, right? So the part where we have cut, that will give the like a gateway for the agrobacterium. Agrobacterium can go in the intercellular space. Okay, so and our motto is just to put them in intercellular space. It's no matter if it is not going inside the cell, it's not required, because agrobacterium has to do its work from outside the cell, not from inside. So our goal is to transfer the agrobacterium, the bacteria, between the two cells, the space between the two cells. So let's assume that it's done, half an hour is done. Afterwards, you need to take out these leaves and dry it. Because it will be wet, and you can't put the wet tissue on this thing. There are chances that other bacteria will grow, even though the medium is having antibiotic. Still, some bacteria, some other fungus can grow. Okay. So to do that, you need to. If you are doing it in a laminar airflow, you need to have this inside. There is a filter paper. You need to have this thing sterile. You need to autoclave it. Otherwise, outside it's fine. But outside will be definitely a transient, because after 2 days, even though the plant will have that gene of interest or not, because it's outside environment, the fungus and other bacteria will start to grow. So you need to take all the stuff out and put it for drying. So when you do this, you will see that one side is dried, another is not, upper surface. So just do like this. Okay, just fold the paper so that upper part will also get dry. So once it is dried, you will see that it's dry. And if you are in a laminar airflow, you need to be very careful, because the airflow will make them dry. So you need them dry, but not to lose its cellular water. So if you put it in a laminar airflow for a longer period of time, what will happen? The air will also dry the cell that you don't need now. So before, and you will feel that the plant is started to wilt. It will like curly in a shape. If it's a leaf, it will get curly in the shape, and you will feel that yes, now it's going, it's dried completely. So you don't need that. Okay, so in laminar airflow, you need to take care of this thing. So usually this plant transformation experiment is done with let's say 500 or 600 leaves, cut leaves. Okay, so you cannot handle 500 or 600 leaves together. You place them for the drying in laminar airflow. So by the time you place the 101, remaining will be completely dry. So you need to put them in a batches. Like if your capacity is 100 leaves, so make the batches of 100 leaves.
आवाज अल्ट्रा
Meanwhile, can anybody say why this technique is more essential? Once we studied in our class, agrobacterial mediated gene transfer.
So this is the experiment of that agrobacterial mediated gene transfer.
Okay, now Sir is explaining about what media. So this is a media, right? This is a media
That is
where
the water
bacteria is
part.
there.
Yeah, bacteria is there.
So bacteria is there in the media. So he cut the leaves, and these bacteria is going to enter into the cut portions. Okay, just have the crude idea what Sir is doing. So why we have to do that? Any idea? Why we have to do that? Why bacteria has to enter the plant cell? Any idea? Why? What delivery? So everybody are supporting GM, genetically modified organism. So we all biotech are now agreeing GMOs is good concept. Free from GMOs are, so it is like we are actually transferring some portion of particular gene of bacteria into the plant cell. Why we are doing it? To create some drought resistant crop, these type of pest resistant plants. That's why we have to use, am I correct, sir? So that is why we have to infect the plant with this bacteria. This is a good bacteria, this won't harm us. Okay, it is also present in the gut. If it is going to the gut also, no problem. So that's why we take the beneficial bacteria and we are doing this type of gene transfer. You should understand every experiment you are going to perform in the next semester, but just have some crude idea. Okay, all the experiment has to be done in aseptic conditions. But just what Sir is doing. After that, we will go to the lab and we have to use laminar air hood. From start, we will say what is laminar air hood, why we should do in laminar air hood, why we should not do in an open situation like this, how much bacteria are there, viruses are there, why we are very specific about using those type of food systems and other things. Okay, please watch it. What Sir is saying, just observe it. Don't get dejected. I am not able to see what is happening here. Everybody will be given chance in our own laboratory, which is we are going to construct. So don't get worried about plant. My focus is on cancer. Somebody is working on Down syndrome. So don't get dejected. I don't want to pay attention in this one. Everything is interconnected. Okay, sir.
So once the leaves are dried, you need to transfer it on the co-cultivation. So this is a co-cultivation medium. Okay. So it's like just like you are handling the bacteria. Okay. So you need to hold it like this. You
They haven't handled.
Okay.
Till now, they haven't handled bacteria, virus, anything. First year is common, so it is.
Okay. You have seen the jars, the tissue culture bottles that are circulated.
Yes.
So in that bottom part, you have seen the medium. So the same medium is here. You can perform it in a petri dish also, but plant cannot. You cannot place the plant here because of the height. Okay, you need more height. You know that plant will grow. So how they will grow here? So, but for the seeds, for the leaf, for the callus, okay, some of the explants are there which can be easily cultivated here. Okay? In case of bacteriology, when you work with the bacteria, you will have the same dish, but the thing is the medium will be different. You will have LB, Luria broth, that kind of media. This is just a plant medium. This is not the medium for bacteria. This is medium for plant. But bacteria can also survive on the plant's medium. Okay. So we are actually cultivating plants, not bacteria. For 2 days, they are waste. Agrobacterium is a waste for plant, but not after 2 days. Okay. So we have to make everything. We have to arrange everything so that your plant cell should grow, not your bacteria. Okay. So here it's the plant's medium. The same, you can see the same color of that bottle and this thing. So it's a plant's medium, but plant medium with some that acetylcysteine and all that chemical also there, so that bacteria. Now we assume that in this thing the plant leaf that is treated with agrobacterium solution, now it has those bacteria, and now we want that bacteria to transfer its genetic material, that plasmid, into the plant's chromosome. So we will take all the care that even though we have cut the leaf, there are chemicals secreted by plants. Still, we are not sure whether the bacteria will do its work or not. So what we will do is we add the acetosayingon inside the medium. So now even if the plant's chemical are not enough to induce the bacteria, the medium is having it. Okay? So usually you can for the bacteriology purpose, it's like this. You can open it like this. Then there is a streaking and all the other process. I don't have that here. It's not applicable. In bacteriology, you need one wire loop. Okay, that you need to streak your bacteria like this. Sometimes you need to spread it uniformly. So for that there is a spreader. Okay, so there is one stage you need to place your petri dish like this, and that can rotate. You can rotate it by your hand. So that spreader, you just put your bacteria here, place the spreader, and then rotate so that the solution of that bacteria will be uniformly distributed, and you will get the colony accordingly. Okay? But this is plants experiment. You don't need to do like this. So what you can do is, let's say this is your this thing, so it is visible to everyone. I am doing it here in my hand. Otherwise, what you can do is like this and just play. Okay. So this is co-cultivation medium. You need to just take the plant like this and place it.
Sir, do bacteria survive in intercellular space even after 2 days, or they dies?
Sorry?
We assume that bacteria have entered in intercellular space. Do bacteria die there, or they survive there after 2 days?
After 2 days, definitely they will have. After 2 days, they grow also.
Yes, sir.
But what we do is we kill them. So
Inside
we will not allow them to grow.
Inside intercellular space, we kill them.
Intercellular space also. It's not that we are placing directly onto the bacteria killing medium.
Okay.
First, we kill them with the higher concentration of that antibiotic. We give the treatment to the plant. First, that is to be done. Let's say if it is getting killed by 200 milligram per liter of cefotaxime. For example, cefotaxime is an antibiotic. So if it is getting killed by that one, what we do is we take 100 milligram per liter of that antibiotic solution, and put our leaf sample in that solution for, let's say, 30 minutes. So within this 30 minutes, the antibiotic will penetrate into that part. It will reach all the cells, including intercellular space, and it kills the bacteria.
Then that our specific gene of interest remain between the intercellular space, they can.
So if the gene of interest is still in the intercellular space, it's our luck. So we gave them two days.
Okay.
We gave the bacteria 2 days, maybe 3 days sometimes. Okay.
Do they not need to cross?
We give them 2-3 days, and within 2-3 days it will transfer. The transfer is like it will transfer to the chromosome, up to the chromosome level.
Like there is cell wall which will prevent external material from entering.
Yes, it will prevent. But see, that's why when I was delivering that talk, I told about the agrobacterium is a natural soil bacteria. Okay, so it has a capacity to infect. If the thing that you are saying is in all conditions is correct, then no plant will have disease.
Yes, sir. Yes, sir.
Because
yes,
bacteria are also active. Bacteria, they are also active. They can also penetrate the plant's defense system.
Yes.
They can do their work after the plant see. And another thing is, this is leaf that is detached from the plant.
Yes, sir.
If the leaf is still on the plant itself will start to react very aggressively
Yes, sir.
and not allow the bacteria to penetrate.
Yes,
But
sir.
now we have taken it out. So now it is on its own. So whatever the defense mechanism it has, it will work with that only. Okay. And for the agrobacterium is not disease causing agent.
Yes, sir.
Although the disease it causes, it says crown gall disease. It causes a disease, but it's not harmful for the plant. Indeed, we can have the advantage of this thing. So that I am showing. Okay. So within 2-3 days, we assume that bacteria will transfer the genetic material to the plant. So now they are no longer needed. So after 2-3 days, what we do is we take this thing out, again put it in a antibiotic solution that will kill the bacteria.
Yes.
Very high concentration of antibiotic that will kill the bacteria. Again, dry them, and this time the drying should be very crucial, because when you go for the co-cultivation, even if it is not dried completely, it is fine. It is still fine. But after 2 days, it should be completely dried. Completely. Because if bacteria actually, if there is a small drop of that left on the surface of the leaf, the bacteria will start to grow. Even though you have placed it in 200 milligram per liter of cefotaxime, it happens. Okay, so all you can eliminate them only through the drying, because bacteria hates this thing, this tissue. So when you put them on this thing, they will not work. So after 2 days, it will be a crucial thing. So this is how it is done. So now after the treatment with antibiotic, after 2 days, you dry them, again place them on a regeneration medium with the antibiotic of selection plus the bacteria killing. Okay? But that cefotaxime that kills the bacteria, you need to put it only up to few days. Okay? Maybe one subculture. After that, you can eliminate it from the medium, because now we know that there is no bacteria. Yes, that's why we have to use the antibiotic which is good for plant but not good for bacteria. But will kill it. There are antibiotics. Okay, like the one that I said, cefotaxime. You can treat your plant in 1000 ppm, 1000 milligram per liter of cefotaxime, and plant will still survive. There is no effect on the for the selection, we should use the kanamycin, because the plasmid contains the resistant gene against the kanamycin. You cannot use this for the selection. You can use the cefotaxime for the killing of bacteria, but not for the selection. For selection, already, if the plasmid has the cefotaxime resistant gene, then you can use the cefotaxime, but there is no gene for the cefotaxime resistance. Nobody is using it. So this is one way, one method of doing the transformation. Now I will show the other method. So see, I told you that we need to cut the leaf so that they can penetrate inside. But sometimes it happens that even after cutting, the tissue is so hard, the tissue is so mature that bacteria cannot go inside. So now we forcefully put them inside by creating a vacuum.
Sir, what kind of plant is this?
I don't know which plant they have taken. This must be the ficus species.
This seems to be bryophyllum, if I'm
Yeah.
Leaf
No,
mesenchyma.
he is asking
Leaf
which
propagation.
species of plant is this.
वो जो बिट्टी कॉटन बिट्टी ब्रिंजल है,
It's outside.
Yeah, it's outside.
बिट्टी कॉटन बिट्टी ब्रिंजल है, उसमें भी प्रोसीडर किया होंगे.
But there are multiple leaflets, so
कुछ लोग कंफर्म कर लो.
growing
हाँ,
from
यही बात है.
single
यही.
Single
leaf.
इसमें तो बैक्टीरिया खुद ग्रो करता है प्लाज्मिड.
Through this
इसमें तो बैक्टीरिया ही खुद उसमें रहता है.
from a
प्लाज्मिड
single
कोई पार्ट
petal
की
there are
बात नहीं
multiple
होती
leaves
है.
that are growing, and the leaf continues to grow from another leaf itself, right? In this plant.
that efficiency is called What is that efficiency called? Okay.
Method is to give the give them a vacuum treatment. So what will happen? Let's say this is the agro solution and there are leaves in it. So first we will create the vacuum. Okay. So while creating this vacuum, you will see that the air in between the intercellular spaces will come out as a bubble. You will see some bubbles here. Okay, if the vacuum is not that strong, you won't see that. But if the vacuum is strong, then you will be able to see that. Afterwards, this is called the pressurization. And afterwards, the next thing is repressurization. So now during the repressurization, all the air that we have taken from the plant, we are giving it back, the repressurization. So in that process, air will not go inside, but the agrobacterium will. Okay, so forcefully they will go inside. So that is a vacuum infiltration method. So let's say this is agro solution and we add some leaf material in it. So we have to first, if I press the if there will be enough pressure, you will see.
The last two pictures, if you're not able to see, you can come and stand here.
Now bubbles have started to come out from this. You can see a few bubbles on the surface. But the pressure is not that enough. It is completely covered. See, air is going inside. So till the air is here, we cannot generate the vacuum.
You
Yeah.
want to create a vacuum.
Vacuum cannot be created. Okay. So otherwise,
We use for filtration.
yes, this is used for filtration. But if we can cover only these things, I can also show that it is also used to create vacuum yeah. So because that is not here, but assume that what happens that the air inside this vessel, you are taking it out to create a vacuum. So first that air is getting out, and then comes the plant part. The air inside the plant cellular space will also come out, and that way you will see it as a bubble.
That's fine, sir. We can.
So during repressurization, afterwards what we do is we turn off this machine and give back the air that we have taken from. Okay. So during that repressurization process, instead of air, your bacteria is going. So this is another method of doing the transformation. First is just treat this thing with agrobacterium. Second with pressurization, repressurization using vacuum. But surely you cannot take this for the tissue culture purpose afterwards. But yes, for the transient transformation you can do. So now we shall go.
Sure.
Okay, so during that time I will